All right, we'll get started and make sure that we share the recording to our community. We're so excited to have our roundtable today with Dr. Hernaz from Hebi and Bio. They've been a tremendous partner for us over the years. Just before we hand it over, we'd like to share that this is a recorded session. So we'll be posting this on our YouTube as well as clips may be used in some of the ways that we've listed here. And if you'd prefer that any parts not be included, just message us and we'll edit those out. And we're really excited for this to be an interactive experience. So please don't hesitate if you have questions to list them in the chat. And if there is any discussion with respect to specific AP heroes in the Q&A, we'll go ahead and remove that as well for the posting in YouTube. So we're thrilled today to have Dr. Harina's bestie. She's a dedicated life science executive and healthcare strategist with a passion for advancing transformative therapies. She has an incredible background in scientific strategy and integrating multiple types of omics, and she brings a unique blend of scientific rigor and business acumen. She's renowned for her ability to drive partnerships and build confidence, especially in innovative biotech and rare disease landscapes. And we're so excited to hear more about the work that she's been doing in AP. All right. Thank you.
Thanks so much, Lauren. Okay, should I go and share my screen?
Yes, please.
Okay, let's see. Can get the slides up. Okay, is that working?
Looks good.
Okay, great. So thank you so much for having me here. I'm delighted to have a conversation really with the Patient Foundation, and also to tell you a little bit about Hebi and Bio, what we do, who we are, and how we've been working with 8P so far. So as mentioned, as Lauren mentioned, I'm Purnaz Behesti. I'm the founder and CEO of Hebi and Bio. At Hebi and Bio, we are a early stage therapeutic discovery startup that is focused on the rare and complex central nervous system disorder space. And so our vision and our mission really is to redefine how neurodevelopmental conditions are viewed, diagnosed, and treated. And we would like to reach a space where basically we can easily match the right medication to the right patient. So a little bit about just our story. I myself am a neuroscientist and a stem cell biologist. I have been in the field of developmental neuroscience for a very long time. I did my PhD in developmental biology at the Institute of Child Health in London at Great Ormond Street Children's Hospital, and then came to the US for a postdoc at the Rockefeller University, where I worked on an autism risk gene. And so that was my foray really into the field of autism and related neurodevelopmental disorders, and I have been engaged in that field ever since. So while I worked on an autism risk gene, which I'll tell you a little bit more about the sort of insights that we gained, and also that formed the basis for the platform that we have developed at Hebi and Bio. I also had the opportunity to help develop reproducible methods for making specific subclasses of neurons from patient-induced pluripotent stem cells in a dish. And I was very much driven by the fact that we realized early on that to be able to really address human disease, we need to have better models of really human biology in a dish that we can actually study. And so while I was a postdoc, I also had the opportunity to be involved in the rare disease world. Initially, I was working as I was sort of contributing in an advisory capacity to a rare disease organization, the FOXG1 Research Foundation. And then I decided to joined them in a more sort of full-time capacity as their first director of science, where I was overlooking the development and really driving the drug discovery and development portfolio for them. And so these interactions, and of course, being in that, in that world, I got to meet many more rare disease organizations, and these interactions and really seeing the patients and interacting with the patients directly have also massively informed the pipeline that we have developed at HeBM Bio. And this was around sort of 2023, mid 2023, when a light bulb went off, where through all these interactions with sort of the patient groups, but also with pharma companies, the very few pharma and biotech companies that are in this space, seeing what is being done and sort of seeing a lot of the gaps. And what I kept on seeing was where the gaps were still in this field very much was really reliable, granular data from patients that was lacking, both on the clinical level, but also at the molecular level. And it was around that time that, we were forming HeBM Bio, that I actually got introduced to Bina through a mutual mentor of both of ours, who himself is he's a CSO for another rare disease organization that's been in this field for a very long time. And, obviously meeting Bina, she struck me as somebody not only very, in it with her heart, which, as a lot of you have obviously noticed working with her over the years, but also extremely astute in terms of what needs to be done at the scientific level, and with sort of like a longer term vision that you really need to have all these important things done really properly from this early stage. And even though everybody obviously is in a rush and wants to get into clinical trials as soon as possible, if you don't do this early stage well, chances of having success in clinical trials goes down, right? So we started discussing a potential partnership and then a small pilot that I will tell you a little bit more about today. So working on induced pluripotent stem cells directly from ATP heroes. But before that, I'd just like to introduce you to the rest of the team that is joining me in this mission. In particular, I'd like to highlight Mary Beth Hatton, who's our scientific advisor. She's a professor of neurobiology, a world-renowned neurobiologist, really. She's a professor at Rockefeller University and also a member of the National Academies. And we have Tal Havochor, who has been advising us and leading the machine learning pipeline. He's a professor at McGill in information systems. And Amir Hosseini Rad, who's really taken on building out the data platform and strategy. So as I mentioned, I've been working in this field for a very long time. And one of the things that has struck me the most is how, the complexity of these conditions. So this is a sort of a graph of neuropsychiatric disorders, but the same is very much true for neurodevelopmental disorders. There's a lot of complexity, both at the genetic level, but also in terms of symptom presentation between these conditions. And we now have identified, so many genes that give rise to neurodevelopmental disorders. Some of them are categorized as rare neurodevelopmental disorders, but actually there's quite a bit of overlap between the rare groups and these more common disease categories that you're seeing here. And importantly, through these interactions that we've had with the rare disease patients, what also stands out is that even in populations where there's a single gene mutation, despite there being a sort of a single genetic cause, the symptom presentation can actually still be vastly different. It can range from anything from somebody who's completely wheelchair bound to being able to walk independently. And so what that tells us is that there's a whole lot going on beyond the genetic level. And again, this is not something that is unique to neurodevelopmental disorders. This is also something that we see very much in neuropsychiatric conditions. And so This level of complexity, both at the genetic level, but also at the symptom level, is a key bottleneck, which leads to poor categorization, which trickles down into ineffective clinical trials. Often there's misdiagnosis, and very few drugs can actually be developed when you have this level of complexity. And so what we are doing at Hebbian Bio is that we have built a data and AI platform where the focus is on really building patient data sets to enable to really get at this level of complexity and to reduce this complexity to lead to better categorization of patients, which will trickle down to enabling several novel applications. And so we have a machine learning pipeline and we have high fidelity cell models. And together we are generating data sets that are really granular and that can enable patient selection beyond the genetics, sort of beyond focusing on just genetics, which will allow us to have better clinical trials, potentially discover novel targets, and eventually be able to also reduce the time to diagnosis, which happens, in many cases can take years instead of days. And so really what we are talking about is a precision approach. And so what we are doing is building really granular molecular data. And for that purpose, data quality, relevance, and granularity matters. So what do we mean when we talk about relevance, right? So if you're trying to address a human condition, the most obvious sort of place to look would be in the patient. So you want to kind of start measuring things in patients, right? So patient cells or patient material, any kind of material is always a much better starting material. However, if there's a neurological component, the most relevant starting material would actually be brain tissue, which is not always accessible. And so what has happened in the field over the past sort of decade or so is that we have now actually started to build models of the human brain in a dish, which allows us to bypass this bottleneck and to actually be able to start with relevant biological material. And so if it's a condition that mostly affects the nervous system, a relevant biological material would be brain tissue and not necessarily blood, which is much more easier to kind of get from patients. So what we have done at Hebbian Bio and also from my previous work at Rockefeller is that we have established really reproducible methods for generating specific subclasses of neurons from patient-induced pluripotent stem cells. And so here you can see some examples of that. So for example, here is a neuron deriving from the forebrain, and these are Purkinje neurons and granule neurons deriving from the cerebellum. And these cells have been repeatedly identified to be implicated in a range of neurodevelopmental disorders. Some fun fact, this neuron here, the cerebellar granule cell, is actually the most populous neuron in the human brain. About 80% of the neurons in the brain are cerebellar granule cells. And so we have established reproducible protocols for generating them in a dish, and we've validated them by checking their molecular profile, sort of benchmarking it against their counterparts in the intact human brain. And to basically start with generating data that is of high quality, relevance, and granularity. Again, another thing that we have focused on is to establish methods for capturing proteins. So while there's a lot of effort to capture what is happening at the genetic level, so genes and RNA, what that information tells you is what cells can do. But what proteins tell you is what cells actually are doing. So it's sort of like a little bit more informative than just capturing RNA or genetic, or at the DNA level, basically. And not only that, but we are also developing methods for capturing protein levels, but also the spatial location of proteins in cells. And again, that's important because that adds a really important layer of information that you wouldn't have otherwise. And it's sort of the difference between capturing an image here that is highly pixelated versus an image to the right. As you can see, it just clearly captures a whole lot more information that you wouldn't be able to get at if you just like look at everything in in a bulk way. So again, this is illustrated in a different way here. So regardless of what is happening at the genetic level, we can generate these protein networks, identify key protein signatures that we then match to patients' symptom severity scores. And that allows us to generate biologically informed precision data sets beyond genetics. That really allows us to kind of group patients based on biology and then enable a whole range of downstream applications, including the discovery of novel targets and biomarkers. And of course, this approach is highly relevant for HP, given the fact that it's a deletion duplication syndrome that covers some 250 genes. And so trying to figure out what would be a viable target is not an easy task. And so the fact that we're looking downstream at the protein level, and we have ways of prioritizing things that really track with the symptoms will allow us to identify novel targets to pursue. And so the rest of the talk, I'm just going to share some of the preliminary insights. And I want to really highlight that this is very preliminary because it's a small pilot, only on 2 patient lines and 1 control. And obviously, this is something that will need to be repeated again in a much larger sample size. But nevertheless, we are seeing some very interesting features that I would like to share with you. So just very briefly, we have taken the stem cells that we've been given, and then we have differentiated or kind of hoaxed them into turning into cells of the forebrain in the cortex. And then we've grown them for about 50 days, and then we've done the analysis on proteins. And we decided on doing forebrain neurons primarily because I have still yet to come across a neurodevelopmental disorder where the forebrain is not involved. So even though we don't actually know exactly which cell types are implicated in HP, and that is work that I know Lauren is actually looking into to kind of figuring out better characterizing which cell types are implicated and what should be modeled. Since we don't have that information yet, we've just gone ahead and done forebrain neurons. And this is also really important because different types of neurons express different things. And so it is important to actually start with something that is relevant. And we can check that we've managed to get forebrain neurons because we can look at markers that are known to be specifically expressed in the forebrain, so things like FOXG1 and PAX6. And then we can also check for markers that are not supposed to be expressed in forebrain, but are expressed in sort of more posterior regions of the brain, such as 801 and GBX2. And also, we can check for specific subclasses of neurons within the forebrain. So for example, CTIP2 is a marker for more deep layer neurons versus superficial layer neurons. And so that's what we are getting. And I just want to tell you a little bit more about, obviously, we are putting a lot of effort into generating high quality data. So for that reason, reproducibility is really important. And this is a technical replicate. You can see a really high correlation between the proteomics profiles between 2 technical replicates. So a technical replicate is basically the same cell line being processed in exactly the same way, but just done twice and run twice. And you would expect if things are running properly and if things are reproducible, you should have a really high reproducibility, which we are seeing here. Other types of replicates, such as biological replicates, are when you start with different starting materials, but you still do the same procedure in the same way. And again, we can actually see that we see quite a high correlation between clones, but also between individuals. And what is interesting, so when you generate induced pluripotent stem cells from patients, or from the blood that has been donated or from a piece of skin. Usually what happens is that you can actually get several clones from the same individual, and it's very common to establish lines from several clones from the same individual. And in theory, those should be, very similar. In practice, they're not always very similar. And so a very common practice when working with iPSCs is to run multiple clones at the same time from the same patient, so that you have a much better reproducibility and representation of what is happening in patient biology. But actually what we are seeing is that there's not that much of a difference between running multiple replicates of two clones from the same patient versus just running different individuals. And so moving forward, and this is something that I've discussed with Lauren as well, I think it just makes sense to just run more individuals in terms of getting better insights into, what is happening at the molecular level in patients. And so in these forebrain neurons that we have, again, despite 8P covering some 250 genes, there's only about 40 of them that are expressed in these forebrain neurons. And again, that is not an unusual thing because not every cell is going to express a full set of all the genes. And here this is more sort of like quality control again. We've looked to see how these genes that we know are on the affected region, how they're behaving. And we can see here, so here are like 4 examples that we've highlighted. And so these 2 proteins are within the duplicated region, and you can see that they are expressed at higher levels in the patients compared to controls, whereas these 2 tend to be in the deleted region. And again, we are seeing lower levels of expression in these. So this is looking promising. And then if we take the whole, like everything that is expressed in the patient cells and compare that to everything that is expressed in the control lines, we can start building sort of pathways, biological pathways that appear to be enriched. So we compare the 2 gene lists, we identify what is differentially expressed, and based on what is different between the 2 populations, we can start building these biological processes. And so if we look at things so you basically end up with things that are down or things that are up. Okay, so if we just look at things that are down in terms of protein level, we can see things like synaptic potentiation and synaptic plasticity, which makes a lot of sense. That synapses, which are these structures in neurons that actually allow communication and connectivity, might be disrupted. We also see axon guidance coming up there as a highly enriched biological group. So these are things that were down. If we look at things that are up, we see cell cycle genes mostly, and things that are involved in chromatin remodeling. And if we look at everything together, again, chromatin remodeling pops up really high, and we still see axon development popping up, and luckily forebrain development, because that's what we were aiming to get. And so basically what this tells us is actually, it looks like, again, this is something that will need to be replicated and reproduced in a much larger sample size, but what it looks like is that the patient cells are potentially kept in a much more immature state because there's upregulation of cell cycle genes compared to more sort of mature markers that are things that drive synaptic development, and also things like axon guidance. And if we dig a little bit deeper in terms of which proteins actually are constituting these biological pathways, if we start seeing things that are sort of like clustered, that's a good sign, because that means that this is more likely to be a real biological signal than just noise. And so we again see things that are implicated in cell cycle regulation, but also ephrin receptor signaling, which is implicated in axon guidance and cell-to-cell connectivity and adhesion. And so one of the more one of the things that we are very excited about is whether we can actually use these molecular signatures, these protein-based molecular signatures, to actually group patients based on really the biology, and whether we can actually see clustering between patients that have more of a sort of overall milder symptom presentation versus more severe symptom presentation. And so what you can see here is a PCA plot of the overall protein profiles of 8P patients, but also another related rare neurodevelopmental disorder, and compared to controls. And what we were happy to see that we can actually distinguish the two different conditions. So you can see that they cluster away from each other. And importantly, the patients that have milder symptom presentation also cluster away from moderate presentation also cluster away from those that have a more milder presentation. And it's also interesting to us that, for example, the milder patients cluster much more closely to a normal protein profile than the more moderate and severe samples do. And so this, once this is done in a more comprehensive way with more samples in there, this is a way to actually molecularly stratify patients and identify targets that could be much more beneficial for the treatment of the patients that have more similar symptom sets compared to sort of like doing one glove fits all approach. So one of the things that we have been building is our machine learning pipeline that allows us to identify targets and prioritize targets. And we actually validated that initially on a existing protein data set from Rett syndrome, which is another rare neurodevelopmental disorder. And we were happy to see that the pipeline was able to identify MECP2 in the top 15 proteins that they prioritized. And we were also able to identify a novel shared target that is actually shared amongst 4 related neurodevelopmental disorders, and is backed by human genetic data. And so this opens up the possibility of being able to actually identify shared targets, not only within existing syndrome classifications, but actually across syndrome classifications, because there's a lot of symptom overlap between a lot of these patients actually. And we ran the same workflow for the protein data that we got from 8P. And this is just to show you very briefly what it pulled up. So again, it pulled up NFM, which not only is it implicated, is it actually sitting in the region that is affected, but it consistently shows up as a protein that distinguishes patients from controls. And it also picked up 2 other proteins that are actually sitting within the 8P affected region. And again, remember, this is amongst basically analyzed proteins. So it's not bad, and it's giving us a lot of confidence that through this approach, we might actually be able to identify candidate targets that we can pursue and we can validate, and that could actually prove to be a good target to pursue for drug development. And finally, another thing that we have been digging into based on this data that we've generated and assisted by AI is to look for existing drugs, existing FDA-approved drugs that are out there and that target some of the proteins that we have pulled up. So, for example, gabapentin is a drug that is routinely prescribed for patients with epilepsy, and it targets CACNA2D1, which is something that we see being disrupted in the patients compared to controls. This drug here is an inhibitor against COMT, which again comes up in the analysis, and it's routinely used for Parkinson's as an adjunct therapy. this drug here, acetazolamide, I can never say it. It is a drug that is very often prescribed for ataxia, and it is not non-selective for CA2, but nevertheless, it does target CA2 as well. And then finally, another drug that we pulled up, FRAX 597. It's a pan-PAC inhibitor, and PAC3 came up as being upregulated. And this is actually in clinical trials, if I understand correctly, currently for another related neurodevelopmental disorder. So these are sort of some of the insights that we can gain with the platform that we currently have. But as I mentioned, for this to really be reliable information, we do need to repeat it. And so in preliminary conclusions, our protein detection assay has high sensitivity and depth. As I mentioned, we routinely identify 9,000 proteins in an unbiased way. What we are seeing indications of is an upregulation, so increase in expression of proteins that are important for keeping cells in the cell cycle, usually, so for cells to kind of keep on growing and not maturing. And we actually see a downregulation, so decrease of markers that are implicated in the establishment of synaptic connections and also establishment of axons that connect neurons together. We have managed to reproduce the result on NfL and NfM, which are not only expressed within the affected 8p region, but also are actually a very robust biomarker in many neurodevelopmental and neurodegenerative disorders. So it opens up the possibility of actually further investigating these as potential biomarkers for 8p. And we are particularly excited about the platform identifying clearly distinguishable clusters based on protein profiles that track with symptom severity. And we would really like to explore that angle much more with a larger sample size. And as I just mentioned, our method of discovery plus machine learning pipeline identified candidate, potentially causative proteins that need to be validated. And the reason we call them causative is because 3 out of the 15 proteins that in the top 15 are actually within the HP deleted and duplicated region. So where could this go next? Obviously, as I've alluded to many times, a larger sample size to confirm preliminary findings and to be able to really lock down key protein signatures that can be validated through other methods and through further testing in patient neurons. I think it's worthwhile considering whether NfL could actually indeed be a reasonable biomarker for HP, and the next step in that would be to look at its expression in blood, in patient blood directly. And then also if the foundation, I know that there's a lot of effort on drug repurposing, but if you are going to ever consider doing a small molecule screen based on the results that we are seeing, we would highly suggest that epigenetic regulators should be included in that screen. And so basically, our vision is by building these precision maps that we can recategorize patients, actually. That's what we really want to do based on biology and not genetics. And we are starting to build these maps with rare neurodevelopmental disorders. And the idea is that we might actually, once this map grows, be able to place patients in these more sort of larger categories of disease categories, and that would completely open up the possibility of more patients, more rare disease patients actually getting included in pharma-sponsored clinical trials, which would be a nice outcome from this. And our really long-term vision for a much better patient journey that currently exists within the space is that, within days of noticing symptoms, you can just walk in, much like how, for example, cancer patients are now walking into the doctor's office and they're just getting a simple blood test done, and through the simple blood test, they're able to get information about their exact tumor subtype. and basically get a biologically informed diagnosis, which allows the doctor to match them to the right treatment for their specific type of tumor. This is how we see the field of neurodevelopmental disorders and neuropsychiatric disorders also evolving by building these precision data sets. And so instead of years, this from symptom to diagnosis to potential treatment, instead of it taking years, it could take days. And I will stop here. My contact information is on here if anybody wants to contact me. The QR code is to my LinkedIn profile, and I'm always happy to answer any questions.
Thank you so much for that incredible presentation. It was so inspiring, and we're so grateful for all of the work that you've done and the bright future ahead.